So good afternoon, everybody. So I also come from a clinical background. So, but a lot of my work involves research. So I am basically an obstetrician, but then I moved on to genetics because I found that field always very fascinating. And when I moved to genetics, I realized that there is so much of genetics even in obstetrics. And many of obstetricians and as clinicians, we are not really aware when to suspect and when do we really do a genetic evaluation for a patient. So my passion is to work on the genetics of pregnancy losses or subfertility, women who keep on losing their pregnancies, losing their babies, or who are having difficulty conceiving. So despite all tests and everything, around 50% of such cases actually still remain unexplained, even in today's era. And we have nothing to offer to them. So I really kind of, that is a field where I want to close the gap and see what could be the genetic factors which could be contributing, and can we use something like, as Dr. Sunita mentioned, that gene therapy is the theme of this session. So moving the next step forward, if we are able to find out that there is something genetically wrong here, could we next step move ahead and use gene therapy at some point of time to help these women? So right now we are not there, but we are at the first step. So what I'm going to show today is the story of a couple who had lost 3 babies, and nobody was able to actually find out what's happening with these babies. And then we were luckily able to do some work. They gave their babies for intensive checkup, and then when we did the genetic evaluation, we were actually able to find out what's happening. And in the process, we actually were able to find a new disease which nobody had reported in medical literature. So this is the story of that couple and how they went through all this process and what happened. So yeah, so this is, let's say, Mr. and Mrs. K. So does anybody know here? I mean, I think you're all from biotechnology background. What are these circles and squares? Anybody knows these circles and squares? Okay. Yeah, so this is, what is this called, this kind of a diagram? Any name is given to this? Yeah, it's a pedigree, okay? It's a diagrammatical depiction of a family tree. So somebody already said these are males and females, so squares are males, circles are females. So this, when they are joined together by a line, horizontal line means they are married. So this is Mr. and Mrs. K, and they have actually had this very poor outcomes in their last 3 pregnancies. So first they lost a baby at 2 months. Then the next time the pregnancy did carry forward, a baby was born at term, but then the baby developed very severe pleural effusion, that's fluid around the lungs. And despite repeated tapping, that fluid just won't go away, and eventually that baby expired on the day 21 of life. And then now in the next pregnancy, this is their 3rd pregnancy now, and in the scan itself, it was now found that there is some fluid around the baby's heart. So that's called as pericardial effusion. So at 23 weeks of pregnancy, because of this fluid around the heart, they actually terminated this pregnancy, and then they came to us and this baby was brought for a test. So what we do in our department is that we are working on these kind of cases, and whenever a pregnancy is lost because there is a problem in the scan or there is an unexplained pregnancy loss, we do an autopsy for these babies, or we do a fetal autopsy. So to find out what is going wrong in the baby's body, and in addition, we also take the baby's DNA sample, and on that we do certain genetic tests to find out the cause. So here now, this 23-week baby, when it was brought for autopsy, so these are the photos of this particular fetus. So to an untrained eye, they may look normal, but we see a lot of dysmorphology in our clinical OPDs in patients, children, adults, and also in fetuses also, which are coming for autopsy. So what is dysmorphology? So if we break down the word, it is dysmorphology. Morphology is the normal anatomy. Dys is something abnormal. So if something is not looking the normal way. So here this baby actually has some abnormality which are very subtle. So there is some mild coarsening of this baby's face, and if you look at the earlobes and the nose, they are slightly thicker than usual. So this, we call it as a kind of coarsening, but there is no malformation in this baby. Then when we did the internal dissection of this baby, so to look at the organs, so that is where we found something very surprising. So this, if you see here, these are the lungs of the baby. The figure which is on the left top, where I've shown the arrows. So that is the right lung of the baby. And on the lung, you can see those bubbles. There are so many bubbles there actually. On the bottom also, on the surface also. Then on the right side top is the liver. Below the liver also, the arrow is showing a bubble. And here again, the pericardium, the left bottom figure around the heart, also the fluid, this is basically another bubble. And in the mesentery, that is around the intestine of the baby, also some bubbles. So there were all these bubbles actually everywhere on the viscera of this baby. And this is something which I had never seen before. And we went through literature and this was never described before. So we were like really perplexed. What is this and what's happening here? So the only thing at that point of time, what I could think is that this looks like maybe because most of the lesions were on the lung. So I was thinking that is it something like a very severe emphysema where, lot of these air pockets have formed in the lung parenchyma? That was the only thought I had. Or I was also thinking that could it be a lymphatic malformation? So there are certain lymphangiectasias which may present sometimes like this, but it was not at all typical for any one of these. So then we thought that we need to investigate. Already previous baby also had something similar. This baby also had something similar. This is probably something genetic here. So the karyotype, which is the basic genetic test, that was normal, both for the parents and for the baby. So we did a DNA-based test. So this is a very advanced test. This is called as whole exome sequencing. So this is basically next generation sequencing, where we are sequencing the coding part of the DNA. So our DNA is made up of coding part and non-coding part. Coding part is just 2 to 3% actually. But that is where the basically the proteins are formed from that coding part. So most of the genetic diseases arise due to defect in those coding regions. So those are called as exons. And if we sequence only the exons, that test is called as whole exome sequencing. So that's what we did here on the DNA of this baby. So when we do whole exome sequencing, usually what happens that you will have lakhs of variants. So that's what happened here also. So then you get all these variants. So around 1 lakh, some 2,184 variants. Then we apply certain filters by checking whether some variant is present in normal population or not, where is it located, what is its read. So like that, finally, in the end, we were able to zero on one variant. Everything else looked okay. There was one variant which was fitting all the criteria, and probably was it matching the phenotype? We are still not sure, but that's what we zeroed in on. So what was this? Basically, this was a variant in this gene, which is called as serpin-A11. So here this was a nonsense variant in the exon 3 of this gene. A nonsense variant means that basically now the stop codon has come so that the protein translation will stop there. So that will cause a smaller or a truncated protein. So this was present in homozygous form, that is both the alleles of the gene were having this particular mutation, and this had a very high CAT score. So then why we thought of this variant? What is this serpin-A11? So actually this belongs to one big family of genes which has some 36 different proteins. This is the serpin family. And the function of this serpin family, they have these different clades, some 9 clades are there, A, B, C, D, E, like that. So this belongs to the A clade, that's why it's serpin-A. So the A clade basically functions as something called as a protease inhibitor. So in our body, there are various proteins. To maintain a balance of these proteins, there are certain proteases. These are enzymes which will break down proteins. So always something will be synthesized, something will be broken down, and there is a homeostasis which is maintained. But these proteases themselves need to be inhibited. If they become overactive, then they will damage all the tissues. They will digest everything in the body. So that is what serpins do. They inhibit the proteases, keep them in check, so that there is a homeostasis maintained in the body. So now this A clade that A clade of serpin, they all function like this. They inhibit some of the proteases. So why were we thinking of this serpin-A11? Because we already know in this clade there is a very famous gene, that is serpin-A1. So that serpin-A1, if it gets mutated, it causes this disease, alpha-1 antitrypsin deficiency. And what happens there is something quite similar to what we are seeing here, but not exactly the same. So what happens there is that there is this protease called elastase. It breaks down the elastin tissue in the lungs. So the serpinase1 keeps it in check. If serpinase1 gets damaged, the elastase becomes overactive and the lungs get damaged. So patient gets emphysema. So that's the problem which happens with SERPINA1 mutation. And that's what we were thinking, that maybe the SERPINA11 is also acting in the same way. That's why the baby's lung has developed these lesions. So, but then we were not still sure, because the SERPINA11, the problem with this gene is that it has never been studied before. So as recent as, this is not working. So till very recent, actually, this gene, till we were working on this paper, it was believed that this is expressed only in the adult mice liver. That's all the information which was there. Nobody knew its structure, nobody knew its function. Some papers even said this is a pseudogene. It doesn't do anything in the body at all. So this is like, we were like, okay, there is no evidence here saying that this gene is working. Why is it causing a disease? We have to see what could happen here. So that's how we started, kind of it intrigued us further, and we decided to move ahead further. So we tried to compare it with the SERPINA1 and see, is it matching, is it similar to that particular protein? So we found that yes, its tertiary structure indeed is similar to SERPINA1. It has this one particular domain which I marked with a yellow arrow, which is called as RCL domain, which is very important for its function.
Go ahead.
So what is happening here with this mutation is that this domain is getting lost, because this domain is formed by 366 to 397 amino acids, whereas this mutation is causing the protein to get truncated at the 224 amino acids. So beyond that, the sequence of the protein is lost. So your RCL domain also gets lost. And this RCL domain is very important for the function of serpins. So which we are assuming now that the function is getting lost. So we thought, yes, it seems like we are onto something. This is probably important. But then how to prove that this is a functional gene? So what did we do next? So till this point, what we thought that yes, there is a mutation, this domain is getting lost, so that means that this protein is not able to inhibit the proteases, and because of which the baby's lungs are getting damaged and the baby is developing all these blebs. So that's what we thought. So we took help of our pathologist. We told him, please take a look in this baby's tissue. Is the tissue really having all these problems? So then the pathologist did a very extensive study of the baby's tissue. So whereby they were able to show that that's the lung tissue, that is the bleb which we are seeing externally on autopsy, which is also seen on histopathology. And in that bleb also there is a lot of disruption of the extracellular matrix of this particular baby. So these are the sections from the lung, and the pathologist was able to show a similar finding also in the liver tissue and also in around the kidney tissues also. So we were able to see that what we were thinking, that yes, this is probably causing a problem because of not being able to inhibit the proteases, we are seeing it in the baby's tissue. So this gave us more confidence that yes, this probably is a new gene or a new disease we are looking at. So we went ahead, we did some more studies. Then we checked literature, so we saw there was one paper where somebody had reported a fetus who also had some fluid in the body, and this fetus also had a variant in the same gene, but they had not done any other studies. They had no other details of this case. So this further corroborated what we were thinking, that yes, this looks like a new disease and this gene is indeed very important in the body. So at this point of time, we counseled the couple. We were still working on the case, but we told them, come back to us when you are pregnant. This may happen again and we can help you by providing you prenatal testing. Unfortunately, they did not really come back in the pregnancy in time. Luckily for them, their next baby was normal. So now they had a healthy baby girl, but then again the next pregnancy, same thing happened. So by 19 weeks itself in the pregnancy, the babies, again, the lungs started developing the same fluid. So they terminated and they brought this baby for autopsy. So this is now their third baby who has exactly the same thing. Again, we can see those blebs on the lung of the baby around the heart of the baby. So same problems we saw in this baby. So we did a genetic workup and we found that this baby also has the same mutation. So the first baby, that is the NGS data which is there, the father and the mother were heterozygous for the mutation, and the second baby is also having the same mutation in the homozygous form. So now, but still all these questions remain. Is this a functional genes? What organs it is expressed? Is it present in the extracellular space? Because the damage is there, what we are seeing. And what does this mutation do? Is it working at level of RNA or protein? Which are the proteases which this particular gene inhibits? So all these questions were still there. So we did further work. We studied, we transfected some cell lines using SDM, and we tried to express the protein, the wild type protein and the mutated protein. And we found that yes, there is a truncation happening there. So it is indeed a functional gene, and this mutation is leading to the protein truncation. Then we went ahead and we studied the mouse tissues by immunofluorescence and also by Western blot, and we saw that this is getting expressed in many mouse tissues, and it has a significant presence in the lungs. So, and the lung is where the baby was having the problem. So then we studied the baby's lungs and compared it with normal, and we found that yes, in the baby's lungs, serpinopathy is not there. Healthy baby, it is there. So with all this evidence, we were able to say that yes, this is a new disease, and this is because of mutation in this particular gene. So this is not a pseudogene, it's a very much functional gene. It is expressed in multiple tissues in humans, in mice, and it appears to be an inhibitory serpin, and it is involved in extracellular matrix homeostasis, and that's what is getting damaged when there is a problem in this particular gene, and that's causing an inhibition or an abnormality in the embryo development, causing this kind of an abortions, recurrent abortions in this particular couple. So with this, basically, we are able to report a new disease, and this is, I would say, a kind of a first where all this work was done completely in India. No other foreign collaborator has been involved in discovery of this particular condition. And then we have, this is found publication, and we have also on the cover of this journal, that is the Clinical Genetics Journal. So these are all the co-authors, all my students, colleagues who have worked on this particular case, and these are my institutes. Nizams Institute, which is a super specialty hospital and Center for DNA fingerprinting and diagnostic, where much of the basic research work was done. So, and then these are the funding agencies through which we were able to do this. So, thank you very much for the patient listening.